Uh, Hello, my name is Jisun Park, and I am a PhD student at the Infectious Disease Research Center of KRIP and Chungnam National University. First of all, thank you so much for giving me this great opportunity to present a global webinar. Today, I would like to answer an important question. How does my research contribute to society? My presentation is titled Beyond Antibiotics, Preparing the Immune System to Save Lives from Sepsis. First, what exactly is sepsis? As you can see from the Sepsis 3 paper on the screen, the major medical societies officially defined sepsis as a life-threatening organ dysfunction caused by a dysregulated host responses to infection. In other words, this means our body's defense system loses control and begins to damage its own organ. This loss of control often triggers what we call a cytokine storm, a sudden burst of too many inflammation signals. The cytokine storm directly leads to multiple organ failing. Currently, our primary defense against these infections relies on antibiotics, but this approach has a critical dilemma. Antibiotics only works against a specific pathogen. They can attack the bacteria, but they cannot fix the body's broken immune balance. Also, vaccines have the same limitation. They are pathogen-specific and take years to develop. Because of these limitations, antibiotic resistance has become a massive global burden, and hospital-acquired infection continue to cause many deaths today. Because fixing a broken immune balance after sepsis is too late, I took a different approach. Instead of asking, how can we treat an infection after it occurs? I ask, how can we prepare the immune system before infection begins? This shift from treating to preparing is the central idea of my research. My goal is to protect the body against both bacteria and viruses, while vaccines also prepare the body, as I mentioned earlier, but they only track on specific pathogen. Recently, scientists have interested in universal vaccines and protein-free vaccines, which are new types of vaccines that do not rely on specific pathogens. This kind of broad proactive immune priming is exactly the defense strategy that my research aims to develop. To do this, we focused on a molecule called DDM and the dodecylbeta-dimaltoside. DDM is non-ionic surfactant, and it is already an FDA-approved ingredient used in other medicines, like diazepam nasal sprays, to help drugs absorb better. This means we already know it is safe for people to use. Because DDM is so safe, our first idea was that it could work as a treatment given after infection. But when we tested this, it did not protect the mice at all. This told me that DDM does not kill bacteria directly either. Based on this finding, I made a key decision. I changed the plan and used DDM before infection instead. This simple change in timing resulted in a 100% survival rate in mice against 3 dangerous multi-drug resistance bacteria, including carbapenem-resistant E. coli, Pseudomonas aeruginosa, and methicillin-resistant Staphylococcus aureus. Furthermore, it significantly decreased the number of bacteria in the blood and organs. To understand how this protection works, I looked at the specific immune cells involved. I found that DDM induced cytokine in mice, which directly increased the number of white blood cells called neutrophils at infection site. To confirm this, I simply removed the neutrophils from the mice and then applied DDM. As a result, without neutrophils, DDM protection disappeared completely. This is clear evidence that neutrophils are essential for DDM to work. To understand exactly how DDM activates neutrophils, first I analyzed their transcriptomic profiles. The data showed that DDM upregulates key defense genes. It genetically primed the neutrophils for effective pathogen recognition and phagocytosis, allowing them to better track the bacteria. Next, the results show that DDM directly activates neutrophils through a specific signaling pathway inside the cell, called GI-coupled GPCR signaling. And this activation switches on the neutrophils. It strongly boosts their production of reactive oxygen species, which is a toxic molecule that kills bacteria, and netosis, which is DNA-based webs to trap bacteria. As a result, these signals drive a highly efficient clearance of drug-resistant bacteria. Because DDM works by boosting the body's own immune system, its protection does not depend on a specific target. To test this, I applied DDM to mice infected with the pandemic H1N1 influenza virus model, and all DDM-treated mice successfully survived. First, DDM induced the production of interferons, then activate the downstream signaling called STAT. Finally, this switches on a group of antiviral genes that effectively block the viral replication. Of course, while DDM works very well, it still has some limitations. It provides 100% protection when given up to 3 days before infection, but this effect is temporary and becoming weak after 7 days. To overcome this limitation, inspired by recent studies, I am currently testing DDM in combination with other immune boosters like TR agonist to provide longer-lasting protection. So coming back to today's question, how does my research contribute to society? I see this in 3 ways. First, rapid clinical repurposing. Because it is already an FDA-approved ingredient in other products, it's much faster than starting a brand new drug. Second, DDM could protect high-risk sepsis patients, especially considering that intra-abdominal infections account for 20 to 30% of sepsis cases. Third, DDM could easily be added into existing abdominal washing solutions used drug surgery, such as taururin. By simply washing the surgery site in this solution, we can directly stop a second infection. Finally, these are our team members, and this paper was published in journal eBiomedicine this year. Thank you for your attention.